Right. Assalamualaikum. Good morning. Right. Can you see my slide? Yes. Okay. Thanks. Right. So we continue our lecture. So today's topic will be LU5, the fifth LU. So we have covered 4 LUs, and excluding this LU, we're going to have 3 more LUs for this course. Yeah. So today's topic is solid state fermentation. So I think in the first LU, I've mentioned in one of the slides, fermentation can be divided into two states. It's either liquid state or solid state. Yeah. So previously, all the things that we have covered are mostly concerned with liquid or submerged fermentation. Liquid and submerged is the same. Sometimes it is used liquid. The term used is liquid. Sometimes the term submerged is used, but it is referring to the same concept whereby the fermentation is carried in the liquid state, whereby you can see that the things are in suspension, okay, in submerged form. So today's topic is about solid state fermentation. So as the name suggests, you can expect that the fermentation is carried out in the solid state. What differentiates solid state fermentation and liquid fermentation is solid state fermentation, it doesn't have flowing water. You cannot see the liquid going through, but it still has certain amount of water. So we're going to discuss that later. So let's have a look at the subtopics that we are going to cover for this LU. So first, we're going to have a look at what is solid state fermentation. What's the definition of SSF? So it's simply known as SSF. And then what are the major factors influence the SSF process? So there are few factors. The first one is microorganisms, yeah, and then the substrate characteristics and pre-treatment. Substrate or the feedstock is the starting material or the food that we give to the microorganisms to start the fermentation. And then the biomass measurement, how we're going to determine when we carry out the fermentation, how we're going to know whether the microorganisms grow well or not. So biomass, it carries two definitions. One is like if you say in the terms of the cultivation or the cell, it can be also said as biomass concentration or cell concentration is the same. But depending on the context you are talking about. So, but if you are talking about let's say the something like agricultural residues, yeah, agricultural residues like the waste or the residues that comes from the industry, from the agriculture, like fruit waste, like from the plants. It is also called biomass because its mass is like something like an entity. Bio is because they are from plants, from living things. So that's why they are called biomass. But in fermentation, we also term biomass as the cell concentration. So you might come across the term biomass concentration, but it refers to actually to the cell concentration. It's the same. Yeah. And then the last factor is environmental factors. What are the environmental factors that influence SSF process? What are the considerations that we must take whenever we cultivate the microbes in solid state fermentation? And then we're going to have a look at advantages and disadvantages of SSF, okay, compared to submerged or liquid state fermentation. And then what are the applications of SSF? Okay, right. So those are the things that we're going to cover. Right. So if you have anything, you just want to, if there is something not working on the screen or you cannot hear my voice, just stop me at any time. Yeah. Okay. So before that, what we're going to talk in this lecture is solid state fermentation in the lab or artificial fermentation. Okay. So I've mentioned before that fermentation, it can happen, it can occur naturally or it can happen, or it can be triggered in the lab, or it can be created in the lab. Okay, so solid state fermentation, if you see it, it is a natural process. If it's not happen in the lab, you can always see it in the environment. For example, if you dump fruit or food waste on the ground, okay, so there are microorganisms in the soil, in the food waste itself. So the solid state fermentation can happen naturally, can happen on those kind of biomass, right? Those kinds are biomass, because it's come from biological things. So those that is as natural as Ssf. And even if you see the composting process when like something if you dump just some of the food waste, after some time you can see that it will turn into black stuff, okay, black things. So that is actually you can even use it as a compost. So composting is a natural Ssf process, but in the lab we also carry out Ssf because we have identified certain microorganisms that can ferment on certain substrates and can produce certain products. Yeah. So the aim of Ssf is the same as liquid fermentation. At the end, we want to harvest the products. So that's why we cultivate the microorganisms in the laboratory. Yeah. And what's the difference between natural and artificial fermentation? Artificial fermentation, we might use recombinant microorganisms. Recombinant are those that are genetically modified. Those that you have altered, you have inserted certain genes so that the microbes can produce certain protein of interest. For example, if you put amylase gene into E. coli, so at the end of the day, the E. coli will produce amylase enzymes, whereby you harvest that amylase enzymes and you want to use it for many purposes, many applications, right? So that is the difference between fermentation in the lab and natural fermentation. In the lab, is actually more controlled, and in some extents, it has been modified. For example, the microorganisms, as well as the substrates. Yeah. So like the substrate in the laboratory, we take care of it, we pre-treat it, we sterilize it. But in the natural environment, who cares, right? It happens naturally. Like the substrates are there and it's not being modified, it's not being sterilized, but still fermentation can happen. Okay, so that's the difference. So let's go to what is the introduction, what is the definition of solid state fermentation. Ssf consists of microbial growth, normally fungi, okay? We talk about it later, why fungi, and product formation on solid particles with minimum amount of water containing sufficient moisture to allow microbial growth, okay? So if you imagine solid state fermentation, it's basically solid state. There's no flowing water or liquid. But it doesn't mean that it doesn't have water. It still have water, because without water, the microorganisms cannot grow. So the solid particles, the substrates, they are moistened with certain amount of moisture, because of the amount of water is very little. So it's normally termed, the quantity is normally termed as moisture, okay? It has certain amount of moisture in order to allow the microbial growth, okay? And how much is the amount of water needed? It depends on the microorganism. Certain microorganisms needs this amount of water, and certain microbes needs certain amount of water, okay? Ssf is a biomolecule manufacturing process, and the product is used in the food, pharmaceutical, cosmetic, fuel, and textile industries. Okay, similar to liquid fermentation. Before this, we talked about liquid fermentation can produce a lot of products that are used in various industries like food, pharmaceutical, fuel, right? And even textile for the enzymes. It's similar thing because Ssf is in general is a fermentation. It can generate product at the end, and the products can be used in various applications in various industries. And one of the, I think these molecules are metabolites produced by microbes grown on the solid media. Okay. So one of the significant application of SSF is what? Did you notice any product that is produced by solid state fermentation in your daily life? Did you notice? Tempeh, yes, good. Yeah, tempeh is an example of kimchi, yes. Any more? Mostly food, can you have encountered food. So tempeh, tempeh, kimchi, what else? Bread. They are examples of products produced from solid state fermentation. Okay, you can see that how significant the process is in our daily life, especially to the food industry. Okay, SSF employs natural raw materials as the carbon source for fermentation, such as cassava, barley, wheat bran, rice bran, sugarcane, cassava solid waste, various oilcakes, corn cobs, sodas, seeds, coffee husks, and pulp and tea waste. Okay, so you can see in general these materials are residues or waste from agriculture. For example, if you harvest sugarcane, and perhaps the peels of it is being thrown. Okay, so that peels, the kulit tu, it actually can be used for solid state fermentation. It's not only sugarcane, but other fruits as well, like pineapple, rice bran. So all of these are actually thrown away to the environment as waste. But people or researchers have discovered that these materials, they have residual sugars or starch. Okay, residual is saki baki apa ni? Saki baki starch and sugar. Yeah, that can be used by microorganisms to grow and to produce products. Okay, so those are the things that people have discovered, I think like 2 decades ago. Okay, it's not new, but it's a few decades ago. People have started using agricultural waste as a substrates for fermenting microorganisms in solid state fermentation. Okay, and these kind of materials or feedstocks can also be used for liquid fermentation. But for liquid fermentation, there are some modifications that need to be done. For example, these stuff are solids, right? So if you want to use it for liquid fermentation, you have to first digest the things and dissolve the things into the liquid, which is not easy, and it requires certain pre-treatment stages. Okay. But if let's say you want to use these materials for solid state fermentation, it's much easier because these things are solids, and the state of the fermentation that you want to carry out in SSF is solid state. So you just have to perhaps clean it, dry it, and cut it into smaller size, and you can put the microbes on it and it can grow on it. Okay, provided that the substrate that you choose is the one that have suitable amount of sugars or starch. Okay, you have do some research on it first. Yeah? You have characterize it, and you know that okay, there are certain amount of glucose that can be used by the microbes. Yeah? So the selection of the substrate or the agricultural residues, it's not every residue can be used. It is based on the composition. It is based on the content. Okay, what kind of composition that are you looking for? It must have starch. It must have sugar in general. And if you see, starch is a polymer of sugar, right? So starch can be degraded into simpler sugar by the enzymes secreted by the microbes. So that's why starchy materials can be used for fermentation. Okay, all right. And if you see, this is not the only materials that are used for SSF. If you see the food industry, tempeh is, what is the starting material of tempeh? You know, any one of you knows how to make tempeh? Soy, yes, correct. Soybean, correct. soybean is the starting material because soybean has starch kan, ada carbohydrates. So it can be used by the microbes, certain microbes that you use, okay, which is Rhizopus oligosporus, yeah, to grow, and then it will produce tempeh at the end, right? Okay. So if you see like different food that you choose, I mean different food that you want to produce, it has different starting materials and different microorganisms. So it's not just like any starting material will do or can work, but it depends. Okay. So these are examples of solid state fermentation. The pictures above here shows the small scale fermentation. Okay, in bioprocess or in fermentation, we do have different scales or different size of cultivation. So the reason is because whenever we start any new project or new research, we have to start with a small scale first. And the small scale is normally termed as lab scale. Okay, so the reason why we don't jump directly to large scale because of we do not know yet what is the best, for the cultivation. And in terms of the material, it would be a waste if, let's say, you don't ferment correctly, and everything will be a waste. So that's why the rule of thumb of research, it must start with small scale, and it will go to pilot scale. The second stage is pilot scale. Pilot scale is the intermediate scale between lab scale and industrial scale. And finally is the industrial scale or production scale. Okay, so so you can see here, like this one is from pilot scale and large scale. So the one that you see above here is the small scale solid state fermentation, where we carry out the fermentation in shake flask. In shake flask, you can see that this one is solid, there's no liquid flowing, and we put this cotton and gauze. This is what we call cotton and gauze. It's not only cotton, but you put it like kain kasar, kain kasar, the one that you use for covering your wound. Yeah, so it's called gauze. Yeah, and then you wrap the cotton inside it, and you tie it, and you use this gauze and cotton in order to cover the mouth of the shake flask. Okay, so even though it's like closed, but actually it allows the transfer of oxygen throughout the cotton. So that's why it is advisable to use shake flask and cotton on top of it for the fermentation. Yeah? And also you can use this kind of setting for liquid fermentation. It's not only for solid state fermentation. For liquid fermentation at lab scale, we also use this kind of setting, like the shake flask with the gauze and the cotton. But this one is liquid lah. So this one is SSF. Huh? So this is a small scale solid state fermentation. In this one is in jar, in bottles. Perhaps this one might be suitable for anaerobic, because if you use closed caps like that or jar, which is not porous, the oxygen might not going through it, and the growth might not be really well for aerobic microbes. So for this one, perhaps for anaerobic. For aerobic, normally we use this shake flask and the cotton and the gauze. All right, so if you see the pilot scale here, it's a bit larger. So just now it's lab scale. You just do it in a shake flask. But once you have optimized means that you have identified what's the best condition for growing the microorganisms at the lab scale, then only then you move to the pilot scale. Pilot scale is a bit larger than the lab scale. So if in terms of the liquid scale, liquid fermentation, pilot scale is something more than, there is a range actually, it's more than 10 liter. Yeah, lab scale is like 10 liter and below, okay? I mean, in terms of the volume. For pilot scale is larger, yeah? And you can see the difference here. In the lab scale, you put it in the shake flask, and in the pilot scale, you use it on a tray because you want to make it you want to have more substrates, and the cultivation volume is more. So perhaps the platform would be different. Okay, this is solid state. For liquid state, you just have like different size of bioreactor, but normally the geometry is still the same. Okay? But this one is a bit different. So you can see that like this one, at the pilot scale, the solid state fermentation is carried out using trays. You put all the substrates, the solid particle, and the microorganism on the trays, and there are lots of trays. And of course, the environment is different, like the temperature control, the aeration is different. Perhaps this one, there is you can see that at the larger scale here, there is a tubing that provides the aeration. Compared to the lab scale just now, there is no tubing or sort of addition of oxygen, kan? So it depends on the natural transmission of air throughout the cotton on the top of the tray flask. Okay, so that's the big difference. It's only some of the differences. It's not all the differences, but you can see that there are many things that we want to consider whenever we upscale, we increase the scale of the fermentation. Yeah. And not just for the solid state, but also for the liquid state fermentation. But in this case, it's for solid state fermentation. So now let's move to what are the factors that influence the Ssf process. So there are a few factors that influence the Ssf process. So first is selection of microorganisms. Yeah. The types of microorganisms. Okay. So what kind of microbes that can grow well on solid substrate, and what are those that might not grow well? Okay, so we know that microorganisms microorganisms comprises of bacteria, yeast, and fungi. Okay, so by right now you should know, if you come across any scientific name, if you come across any fermentation process, you should know that fermentation is a bacterial or yeast or fungal fermentation. Okay, so that is something that you should know by the end of this course. Okay, so basically, all types of microorganisms, bacteria, yeast, and fungi, they are bacteria, they are yeast, they are fungi that can grow on solid substrate. Okay, but basically, the one that is the most favorable is fungi compared to bacteria and yeast. If you see bacteria, yeast, and fungi, the size is increasing from bacteria to fungi. And also the morphology is different. Morphology is the characteristics, the physical characteristics. Yeah? If you observe all those microbes under the microscope, you can see different morphology, right? Different features, different appearance. Yeah? Morphology is appearance, physical appearance. So let's move to the next slide, because I need to show the fungi. Okay. So normally, the reason why fungi is more suitable for solid state fermentation is because fungi have this sort of filamentous structure. Okay, filamentous means the one that are branches. If you see here, these are all the types of fungi: Penicillium, Rhizopus, and then Aspergillus. So filamentous ni dia macam bercabang. It's not just like single cells, right? So those feature or those characteristics is beneficial, because if you think about the solid state just now, it's not in free-flowing motion. So solid state, for example, if you use fruit peels, everything is in solid state, in static condition. So how you want to make sure, if let's say you use, if you want to choose what kind of microbes, it must be the one that can actually grow and reach the microbes by itself, reach the substrates by itself. Meaning to say that it must have those kind of branches. Okay, so if you imagine bacteria with the single cell kind of characteristics, it might be difficult for it to reach the substrates, right, throughout the fermentation. You got what I mean? So that's why filamentous fungi are the best and the most common organisms used in solid state fermentation because of this structure. Okay. Advantages of using filamentous fungi for bioconversion of solid substrates, the hyphal mode of growth. Okay, so throughout the time, if you incubate the cultures, throughout the time, the filamentous, the structures of the branches, it will actually grow longer and longer. So it can reach more substrates throughout the time, more and more. Okay? So compared to if let's say it's just static at one point, like the bacteria, because the bacteria doesn't have the filamentous structure. Okay, so another important point why fungi is more common for solid state fermentation is because fungi have high tolerance to low water activity. Okay, low water activity. So solid state fermentation is without flowing water, right? Free flowing water. It has water, but very little, very minimal. And fungi, if you grow it, in other words, it is actually more robust than bacteria. Bacteria needs more water, whereas fungi may need little amount of water, so it can survive in a situation or in a state that do not have much water, like in solid state cultures. Okay, so it has fungi have high tolerance to low water activity compared to bacteria. And then fungi have high osmotic pressure conditions. So whenever the things are in solid, perhaps at certain points it may create certain pressure inside the cultures because it is not flowing and it is like being confined to one place. So fungi can resist high osmotic pressure compared to bacteria or yeast. Okay, but it's not that there is no bacteria or no yeast that can be fermented on solid state substrates, but there are a few, not as much as fungi. Okay, still on fungi. The hyphal mode of growth gives the filamentous fungi the power to penetrate into the solid substrate. Therefore, nutrient can be utilized by fungi efficiently. Okay, so I've mentioned about it, right? So if you see this one, hyphal growth from spore. So perhaps if you, and I think you have known already before, right? Fungi have longer incubation period for fermentation compared to yeast and bacteria. Bacteria, the doubling time is just few hours. So by 24 hours, it already reached the death phase. Compared to fungi, it needs maybe 2 weeks in order to complete the growth curve, the lag phase, log phase, stationary, and the death phase. Okay. Right. So in terms of the growth of the fungi, so what is hyphal? What is filamentous? It's something to do with, as I mentioned just now, the branches. So throughout the time, okay, from day 0, if you see this small box over here. So if you cultivate the microbes from day 0, it might look little as this. The first one here, if you see the dot and the single branch, single line over here. So it may start with that first, okay, on day 0 or day 1. Okay, so throughout the time, it will grow, and the branches will be added, right? It will grow. I mean, the branches will be increased, the number of branches. So if you see here, the growth of it, the germinating spore, it will be more in the 3rd picture. And in the 4th picture, you can see that there are lots of branches. So you just imagine if you grow the things, if you grow this small dot, this initial amount of fungi on a solid substrate that is static and not moving. Yeah. So you grow it there, and after some time, it can actually reach a lot of spots or points on that substrate because of the mycelium structure. Okay, so I hope that will help you to understand why fungi is more suitable and more effective to be cultivated on solid state substrate compared to bacteria. Okay, so bacteria, if you see bacteria doesn't have those branches kind of things. So it only have like small dots. Yeah, so throughout the time, it might be maybe more. It can, I mean, it can increase in numbers, but in terms of the structure, it's still circle, right? It's still dot. So it's very difficult for the bacteria to actually reach other parts of the substrate, yeah, in the solid state. Okay, so make sure you understand what is this hyphal mode, this kind of things. Is a hyphal, the branches like in plant language, but in scientific terms, we call it like hyphal mode, mycelium, okay, filamentous. Those are the morphology of fungi. Okay, this is another picture showing how. Okay, this one is, if you see, this one is, okay, this one is the substrate, yeah, the food. Substrate is the food, yeah? Substrate, and then this is the filamentous fungi. Okay, so you can see that this one, if you see this one, is a cross section of it, and this is the surface. Let's say this is the substrate. These kind of things are substrate. So that's you can see that the hyphae can penetrate into the substrates, yeah? And it's not only at one point, but all parts of the substrate, yeah? Okay, so once the hyphae, once the structure of the fungi penetrates into the substrate, that is when the enzymes are secreted, okay? So because the microorganisms, they are the enzymes. So those enzymes, they are actually degrade macam like polymers, like starch into sugars, so that the sugars can be used by the microbes to produce products at the end, okay? So I think I've explained this as well just now. So this is how the things looks like, how the penetration of the hyphae into the substrates. Okay, so here are some examples of microorganisms and the Ssf process, and also the products actually. So they are, if I mentioned just now, they are bacteria that can be fermented, but very limited. There are also some yeast, but also not as much as fungi. And it's not just this, actually the list is long, but this is just some examples, okay? And what determines the types of the products is the type of the microorganism. Aspergillus oryzae is used for making koji. Koji is a Japanese food, for making citric acid as well. And then Aspergillus niger for production of feed, proteins, amylase. It depends sometimes. Some of the microorganisms can be genetically modified. It carries certain gene of interest and it produces certain product of interest, okay? Rhizopus oligosporus is the one that is used for making tempeh, yeah? Soybean. So you take the Rhizopus oligosporus and then you put it on the soybean. At the end of the day, you can have tempeh. So if you put Aspergillus niger on soybean, it might not produce tempeh at the end, okay? So the selection of the microorganism play an important role in determining the types of the product produced. And not just the microorganism, the substrate, okay? So if you grow Rhizopus oligosporus tadi kan, not on soybean, but on something else, like apa ya, like other things lah, maybe on flour ke, kan? So it will not produce tempeh at the end. So you can see that how important the type of the microorganism and the type of the substrate, okay? And not just the type of the substrate, is also the conditions, the pH, the temperature, the environment, yeah? So those kind of things are the one that we manipulate. So that's why the fermentation in the lab is the one that we control. We control everything about the process, yeah? Okay, so some examples of bacteria, Streptococcus for composting. Composting is the production of compost. Bacillus for amylase. Saccharomyces cerevisiae for food. For food, this one is for bread. For production of bread. And for production of ethanol. Ethanol is more common for liquid fermentation, but they are solid state fermentation that can produce ethanol. So if so this lecture is recorded. So if let's say you have difficulty to join, okay, so you can always refer to the recorded lecture that I will post on eLeak and YouTube. Okay, so don't worry if you I saw some of you quite maybe you don't have good internet connection, but don't worry about that. Okay, it's recorded, so you can always refer to the lecture later. Okay, so any questions so far, guys? Any question? I need some response from you from time to time, just to make sure that I'm not talking alone. No? Okay. You understand what I've explained? Okay, thanks. No question. Okay. Means that I assume that you understand what I have explained. Fermentation is not really a difficult topic. It's something that you can always relate to daily life compared to genetic engineering. Lagi susah sebenarnya. So this one is, that's why I like fermentation field, because it's something very easy for me to understand and very easy for me to relate to daily life. Okay, so having said that, I hope that it's not difficult for you guys to understand. Okay, so anytime if you don't understand, you just stop me at any time. Okay, so what are examples of filamentous fungi in Ssf? Okay, let's have a look at some examples. So in the food industry, there are so many. It's not just the one that we have mentioned just now, but there are actually so many types of products, food produced by solid state fermentation. And even there are some that I might not know even, because it might be local food. Contohnya, we know some of the food like tempeh because is produced in our country, right? So that's what we know. But other foods like in Germany, they also have their sort of fermented food. There are so many actually. Okay, so one example is koji. Koji rice is a Japanese food. It's a cooked rice that is inoculated with Aspergillus oryzae. It's a fungi. It's a mold. It's also named as koji mold, the local name. The rice is then placed in wooden tray, left to ferment in warm and humid environment for up to 50 hours. And the result is moldy rice. Okay, so moldy rice, if you see this one here, this is example of koji rice. It's not common in our country because perhaps we don't eat this, but it's very common in Japan. It's their local food. So if you see the description here, it has Aspergillus oryzae. Okay, it eats the cooked rice. Cooked rice has what? Starch then. So that starch becomes a carbon source. Carbon source to the Aspergillus oryzae. And at the end, they can produce certain types of perhaps organic acids, moldy rice, in the moldy rice. So that's why sometimes you see the kuih tapai, dia rasa masam kan at the end because it has some organic acids. Okay. So here you can see also the environment plays an important role. So it plays in wooden tray. So wooden tray ni bila dia dalam tray, so it is exposed to air. So that's why dia bukan dalam jar. Certain fermentation or certain, if let's say you make kimchi, I think it's fermented in a jar, but this one is apa ni? Dia needs aeration perhaps. So it's best to be placed on the tray, right? And then the environment is a warm, humid environment. Perhaps the temperature is a little higher than the room temperature, and the incubation period is 50 hours. So the process is under certain temperature, on certain platform, and during certain period. So those are the parameters involved in any fermentation actually, but in this one is specific to the koji rice. So what happened during the fermentation? The fungi digest starches and proteins and breaks them into sugars and amino acids. So why starch or sugars are needed by the microorganisms? Because they eat that. That are the carbon sources. Same as humans, we need rice, we need bread, right? As our carbohydrates, carbon sources. So the same goes to those microorganisms during the fermentation. And koji rice is used as a starter for a number of fermented Japanese food products, including sake, soy sauce, and miso. Okay? So those are Koji rice. And then in Southeast Asia region, in Indonesia or in Malaysia, what is common to us is tempeh. Tempeh is made from cooked soybeans, where Rhizopus oligosporus is grown on it, right? And then after a certain period of time, you'll get tempeh. Okay? But the process, the fermentation process, dia pun ada dia ada certain conditions lah, like at 30 to 35 degrees for 1 to 2 days, the period. And apa lagi? The pre-treatment of the soybean. I think you have watched. I hope that you have watched the video that I posted on Elite, the preparation of tempeh. So you have to do something first on the soybeans, right? You have to cook it. You have to soak it. You have to dehull it and then cook a soak, and then cook the soybeans, allow to cool, and only then you put the starter culture, which is the Rhizopus oligosporus, and then you incubate for 1 to 2 days at 30 to 35 degrees, and at the end you'll get the tempeh like this picture. So the white thing here is the mycelium, the filaments of the Rhizopus oligosporus. Okay. But this one is edible. This kind of yeast are not harmful. That's why you can eat tempeh. Although you know that they are actually the mycelium of the Rhizopus oligosporus. Yeah? And then we have tapai. Okay, tapai, kueh tapai. So kueh tapai is made from cassava, white rice, or glutinous rice. You can use it as the substrate, as the starting material that you feed to the ragi. So normally we use ragi, right, for tapai. What is ragi? Ragi is a local name, but actually in ragi there are many microbes. Actually, it's a consortium of microbes. For example, Aspergillus oryzae, Rhizopus oryzae, and yeast including Saccharomyces cerevisiae, along with bacteria. So ragi, I cannot say one scientific name, but it's a consortium of many types of microbes. Yeah. And if you want to know what are those specific microbes, you can do research on it. You just can take the ragi and then you analyze what's the microbes in it. But basically, the examples of microbes are Aspergillus oryzae, Rhizopus oryzae, and Saccharomyces cerevisiae, and also some bacteria. Okay, so So how you make the tapai? You cook the rice first, and then you put the ragi on it, and then you ferment it. When you ferment it, you keep it at certain place and for a certain period of time, and at the end it will get the tapai with tapai then. Okay, but I think there is a certain period of time. I'm not an expert in making tapai, but there are a certain period of time that you have to take note, because otherwise, if the fermentation is too long, it will produce ethanol as the end product. So it will be alcoholic. Actually, the tapai ni pun I rasa dah ada juga certain amount of alcohols, but dia tak sebanyak if let's say you extend it for, if you extend it above certain period, it will be more alcoholic. Okay. So that's the selection of the microorganisms. Okay, you can see that how important the microbes, the selection is, because it will determine the type of the products produced. We move to the second factor, which is the substrate characteristics and pre-treatment. Okay, so if you see the substrates used for the food just now, they are actually originally food, right? Like soybeans, the rice. You ferment it and then you get different products at the end. But in other types of Ssf research, people have moved towards using biomass around us, like the agricultural residues, the industrial residues or waste, right? So those are the things like shown in this picture. If you see this one is I think the hampas from sugarcane, kan? sugarcane, and this one I think from different sources. Okay, but of course you don't use these substrates for making food. So these substrates are used for fermentations that produce products that are not to be consumed by human, not like food. Okay, it's like for making enzymes, for making fuel. Okay, that's fine because you don't eat it. Yeah, so it's from the waste convert to fuel, so it's fine. Yeah, so those are sorry. So those are the substrates, yeah, that can also be used as those are the materials that can be used for substrates for solid state fermentation. Okay, but let's have a look at what kind of when we want to choose the type of substrates that we want to use for fermentation. First, we have to know what are the composition of the substrates, whether or not it has sugars, whether or not it has starch that can be used by the microbes. If it doesn't have starch or sugars, still it is not useful. It cannot be used for the fermentation. Okay, so the composition plays an important role, and then it must be porous. Okay, I just go like important points. I don't go word by word. It must be porous. What is meant by porous? Porous means that the substrate can be accessed. It can be. It's not. It can be penetrated. Porous, something that can be penetrated by the microbes in order for the microbes to get whatever the sugars or the starch inside the substrate. Okay, so that is one of the important characteristics of the substrate for fermentation. Okay, and then the substrates should not be contaminated by inhibitors or of microbial activities, or and should contain, okay, I mentioned about the composition, sugars, and apart from sugars, apart from the carbon sources, it must also have nitrogen sources. I forgot to mention about it. And also mineral salts, because those are actually become the food to the microbes. But the point that I want to emphasize here is that it should not be contaminated by inhibitors. So it means that the substrate is free from certain types of inhibitors or contaminants that can interfere the growth of the microbes. Okay, apart from it must have a certain amount of carbon sources, like sugar and starch, nitrogen sources and mineral salts. It should not be contaminated by the inhibitors. Okay, so those are the things that help the selection of the substrate for fermentation in general. This is not actually for liquid state fermentation. Also, we can employ the agricultural residues, but the preparation is different. But now we are focusing on solid state. But to say that actually the agricultural residues can also be used for the both liquid state and for solid state fermentation. Okay. And what are the other things that we need to also consider concerning the substrate of the fermentation? Okay. So if you see macam contoh yang senang yang tengok tadi kan, hampas tebu kan. So if you see those kind of structure, it's quite long, and then the size might be varied. There might be short hampas, ada yang panjang. Yeah. So before we start any solid state fermentation, when we want to use the agricultural residues, it is very important to do some sort of pre-treatments. Yeah. So what is meant by the pre-treatment is how we're gonna prepare the substrates so that it will be used easily. It can be accessed easily by the microorganisms. So the pre-treatment involves several necessary steps, such as size reduction. Okay, so it's one of the example of the pre-treatment, meaning to say that if you have fruit waste, what you have to do first is perhaps you have to dry it. And then once you dry it, you have to reduce the size into uniform size. And normally in the lab, we normally use powderized form of waste or substrates. So you grind, let's say the residues that you have chosen, you grind it, and then you chop it into small size, and perhaps you blend it, grind it, so that it at the end it will be in a powderized form. Okay, so the reason why the smaller the size, it will be better because it will be easier for the microbes to assess it. Okay, to assess the substrates throughout the fermentation. So that is the purpose of reducing the size. And then physical, chemical, and enzymatic hydrolysis of substrate. So what is meant by this? It depends on what type of substrates that you are working with and what type of microorganism that you want to ferment on the substrate. Okay, so if let's say you know that your fungi do not have certain enzymes yang boleh degrade starch into sugar. Okay, so meaning to say that you have to do those hydrolysis of starch before the fermentation. Okay, so that is to be done by these steps. Hydrolysis of the substrates. So hydrolysis, it can be done by physical hydrolysis, by chemical hydrolysis, or by enzymatic hydrolysis. So physical hydrolysis, it can be you can use certain physical treatment like maybe macam apa ya, sonication, the physical process lah. And then chemical process is like you add some chemicals like acid or base in order to break, like for example lignin. Contohnya, lignin tu ada satu komponen dalam biomass. So when you break the lignin, and only then you can assess the cellulose or hemicellulose, whereby those cellulose can be degraded into sugar at the end, glucose. So if let's say you don't break the lignin, you cannot get the cellulose. So that's why you have to treat first with acid or base, depending on the types of the substrate, so that you can get the that sort of cellulose. Okay. Enzymatic heterodysis is when you add certain types of enzymes that you know can degrade whatever the things that you want to target on the substrates. Okay. Right. Supplement with nutrients. Okay. So those residues, sometimes they just have certain amount of, let's say, sugar, tapi tak ada nutrient lain. Contohnya nitrogen sources. So in order for the fermentation to grow well, for the microbes to grow well, so you have to add some sort of nutrients in order to supplement the substrate, the media. So perhaps if let's say you know that it has sugars, then you add some nitrogen. Kalau tak ada nitrogen lah. Okay. So that is supplementation with nutrients. It can be phosphorus, it can be nitrogen, it can be salts, depending on the process. And then controlling the pH and moisture content. Okay. So before you start the fermentation, what you have to do with the substrates is that you have to first adjust the temperature, adjust the pH, temperature masa cultivation, pH, and also the moisture content. Okay, pH depends on the optimal pH of the microbes that you want to ferment. Moisture content, you add some water into the, let's say you dah ada powderized sago hampas, for example. So you add some water to it, but you have to know certain level. Okay, so normally we measure the moisture content in terms of percentage. Let's say 60% volume per weight, for example. Okay, so it's measured in terms of percentage. And then cooking or vapor treatment for macromolecule structure, pre-degradation or elimination of major contaminants. Okay, cooking ini tadi just now, if you prepare the soybeans for the tempeh, right? For the tempeh production, you cook the soybeans first. So that is a kind of pre-treatment in that particular process. You cook the rice. Itu pun salah satu pre-treatment for that particular fermentation. Okay, so that's the pre-treatment. And the importance why we have to do the pre-treatment of the substrates before we start the fermentation. This one is before the fermentation, before you add the microorganisms. The fermentation does not happen yet. You have to prepare first the substrate before the fermentation. It's the same thing macam you dekat dapur lah, before you nak masak kan. You have to cut the vegetables into smaller size, and you have to clean it. You have to like chop it. So it's the same thing like fermentation. We have to do some sort of pre-treatment before the fermentation. Okay, the most significant problem of SSF is the high heterogeneity of the substrate materials, which may lead to non-uniform substrate structure, variability between batches of substrates, and difficulty of mixing solid mix, causes non-uniform growth, temperature, and pH, and etc. So this is one of the among the problems related to SSF, because the culture is in solid state, so tak ada, like unlike the liquid, you can always stir it, but for the liquid, it's very important for the to maintain the consistency or uniformity of the cultures. So because of these problems, that's why the pre-treatment that I mentioned above is needed. Okay, so that's why you have to reduce the size in order to enhance the fermentation process. Make it into like the smallest size possible, like the powderized form, compared to if you have, let's say, the one that is not chopped, the one is not cut into smaller size. It would be harder for the microbes to reach the substrates. And even if in the powderized form, it is still actually not easy for us to make sure that the microbes use all the substrate supplied. So still the uniformity problem is still a problem. But it can be reduced if you do those pre-treatments. All this pre-treatment. Okay, so that's the second factor. The third factor is the biomass measurement. Okay, so you know now that the state of the solid fermentation with the fungus, especially fungus is the normal or the usual microorganism used for the SSF. So you can see that the whole thing is actually in static condition, and it's actually hard. Actually, it's actually difficult for us to determine how well the growth of the microbes. Okay, so that is actually one of the challenges in SSF, which is the determination of biomass concentration. So biomass here refers to the cell concentration. Okay. Direct determination of biomass in SSF is very difficult due to the problem of separating the microbial biomass from the substrate. Okay. However, absolute amount of biomass is important for calculation of growth rates and yields. So although it is difficult to know like how you're going to quantify, it's a bit tricky, but somehow we need to actually measure. We need to know how well the growth of the microbes, how well the growth of the fungi during the fermentation. Okay, so because without monitoring it, we do not know at the end whether we are fermenting for nothing, or is there any product from the fermentation, because the fungal fermentation is long. It's like more than a week sometimes. Usually it's more than a week. It's 2 weeks. So we don't want to waste our time waiting for nothing. So that's why the monitoring is very important throughout the fermentation. Okay. So how are we going to determine the biomass growth or the cell growth in SSF? Okay. So normally it's by using microscope. Using microscope. So you take certain amount of the culture and then you observe it under the microscope. And normally we use scanning electron microscope. So this is a picture of fungi under scanning electron microscope. You can see the structure of the branches of the filaments. Okay. Although it is possible to monitor or to observe the morphology, but it's still actually difficult for us to quantify how much the growth from, let's say, from day 0, day 4, day 8. It depends on, you have to analyze by yourself how well is the growth, and yeah, perhaps it can be in terms of the qualitative measurement, but in terms of the quantitative measurement, it's a bit tricky because you cannot really like measure exactly, you know, like the dimension of the filamentous under the microscope. Unlike submerged fermentation. Submerged fermentation, it is in the form of liquid, right? So the sampling throughout the fermentation period, you take certain amount of liquid, you measure the absorbance using a spectrophotometer, and you can see if, let's say, the cultures grow well, you can see the increasing trend of the absorbance. It shows that the culture grow well. So you can quantify the growth, right, easily using the liquid cultures, liquid fermentation. But it is not that really possible for solid state fermentation. So what you can do for the solid state fermentation, you can observe the morphology of the fungi or the microbes that you grow, and you must actually have certain understanding, like what is the how is the good growth looks like. Okay, you must have certain understanding on it. So that's why I said it's more to the quality of the measurement or monitoring, rather than quantitative. Quantitative study absorbance you can see increase from 0.1, 0.5, 0.8. So that's quantitative wise. But qualitative wise is something that you have to analyze by yourself. You have to see the trend, the traits or the features based on your prior understanding about the morphology of the fungi. Okay. May I ask, did coffee fermentation? Oh, I missed the ni. Apa soalan dia? Did coffee fermentation using submerged fermentation and using absorbance as measure? Coffee fermentation, kalau you tengok proses, the process of making coffee is actually solid state, kan? Sebab dia actually dia di ferment. It depends juga lah, I think. I bukan, it depends on the industry as well. Tapi yang what I know, the traditional one, it use solid state fermentation, okay? Whereby bila dia solid state fermentation tu dia akan dia actually perap kopi tu untuk dapatkan apa ya dia punya tu, that sort of ni lah, at the end dia dapat to remove the mucilage, I think, something like that. I think I posted this on Instagram, so you can refer to Instagram, the coffee fermentation. The classic one is using solid state, tapi I'm not sure about the current trend in the industry. Perhaps they are submerged fermentation. Okay. So dia actually dia tak ada specific one product tu, hanya except for tempeh lah, macam tempeh is very obvious, macam bioethanol kan. It can produce by submerged fermentation. It can also produce by solid state fermentation. So there are certain products of fermentation that can be produced by both methods. Macam enzyme pun sama. Enzymes, there are enzymes that can be produced by solid state. There are enzymes that also can be produced by solid state submerged fermentation. Contohnya amylase. Amylase boleh diproduce guna solid state, boleh juga diproduce guna submerged. It depends on the process, the technology, and also what is the scale ataupun the interest of the process. Okay. I hope I answer your question. Using absorbance as measurement. Okay, submerged, kalau liquid fermentation, of course, you can measure the absorbance. Is a method for measuring the growth. Okay, so because when you measure the absorbance, you translate it into the cell concentration based on the standard curve. So that's why I put it in the practical with the questions, because those are the things that you have to apply. What is actually how you want to what you are going to do in the lab concerning the fermentation experiment. So, and absorbance kind of thing is not a new thing for you, right? You have studied how to measure absorbance in biochemistry, practical, and also what is dilution factor. And then you have to know that absorbance ni dia hanya, the range is from 0 to 0.9, the machine. Kalau you measure more than that, it means that your reading is not accurate. So what you have to do, if you have concentrated samples, you have to do dilution. right? So that is the purpose of dilution. And what are you going to do with that? So if you do dilution, I think you have studied this in biochemistry, how to do dilution. Dilution factor, what is meant by dilution factor? And when you measure the absorbance of the diluted sample, you have to take note the dilution factor as well. And what you're going to do with the dilution factor, you multiply with the absorbance of the diluted sample, okay? And that will give you the actual value of the absorbance. So dia memang at the end you akan dapat absorbance value after you multiply with the dilution factor. It can be more than 1, like 3.1 macam tu. Tapi it is after the multiplication with the dilution factor. It's not the reading that you get directly from the machine. Okay, the machine only reads absorbance within the range of 0 to 0.9. Okay, if you got more than that, you have to do dilution. Okay, and you are going to do this, you apply this in your FYP. If you're using spectrophotometer, make sure you apply the right technique. Otherwise, if you don't dilute your samples and the samples, the machine may give you a reading of like 1.2, but it's not actually the right. Dia dah pening dah sebenarnya. Dia punya machine tu dah tak, dia bukan range dia dah. Okay, actually tu dah tak accurate. Dia bagi reading let's say 1.2. Tapi sepatutnya kalau you do the dilution, you measure the diluted samples, you multiply with the dilution factor, you might get the reading of 3.5 macam tu kan sepatutnya. So you can see the error that you have made, 3.5 compared to 1.2. So that's why you have to apply the right technique in measuring the absorbance. And I have put it in the practicals in order to give you some exposure, even though I know that you have been exposed to it in your biochemistry practical. Okay. And in bioprocess, those who are doing FYP, if your topics is under fermentation, you're going liquid fermentation, you're going to use spectral a lot and make sure you apply the right techniques. Okay. All right. Let's continue. Mana kita tadi? Oh, about this one, right? Direct evaluation. Okay. So for the solid state fermentation, how we're going to measure the biomass, how we're going to know whether our microbes grow well or not during the fermentation period. One of the options just now is like by observing under the microscope. Okay, that's one of the ways. Another way is by measuring the metabolic rate. Okay, for example, the oxygen consumption, carbon dioxide release. Okay, oxygen consumption, if let's say the Ssf is aerobic, if the process is aerobic, meaning to say that it consumes the oxygen. Okay, so this is actually more common for large scale Ssf. Okay, so for example, this one, if you see the machine over here, this is a drum bioreactor, whereby it is used for conducting solid state fermentation in larger scale, perhaps in pilot scale. Okay, but if you carry out the Ssf in shake flask, it's not possible for you to measure the oxygen consumption. Okay, so this is the limitation of this method. It's only applicable for reactor that have the oxygen or the aeration inlet, whereby we can measure, we can know what is the uptake. Yeah? Okay, so if let's say the oxygen is consumed, this is indirect method, right? So in the first LU, I've taught about different measurements or different ways how to measure the growth of the microbes, direct and indirect method. So this is under indirect method. So if let's say it's for aerobic cultures, if let's say the oxygen is, we know we can observe this kind of machine or reactor, dia ada inlet for the oxygen, for the aeration. Yeah? So, and you can, the recent or the modern bioreactors, it can give you the profile like this on the screen and on the computer, yang modern punya fermenter. So you can see that the trend of the oxygen, whether or not it is static at, like if you provide it at certain percentage, normally oxygen ataupun air ni kita provide in terms of percentage. Let's say the percentage is, the dissolved oxygen is about 90%. our initial feed. So if let's say that 90% is reducing over time, it means that the oxygen is being consumed, right? So it gives an idea that the microbes inside the reactor are using the oxygen to grow. Okay, so that's how we can quantify and we can estimate the growth lah. I mean, in terms of, we can monitor. Okay, so the microbes are growing because it consumes the oxygen. Okay, so that's why it's indirect method. It's not directly, but it can be used as an estimation or approximation about how well the growth of the microbes inside the reactor. Okay, and then also apart from the oxygen consumption, carbon dioxide release. If let's say you know that the process, your fermentation process produces carbon dioxide as a product. Okay, so if there is a rise in the carbon dioxide, it means that there is a fermentation happening inside it. Okay, so you can assume or you can approximate the carbon dioxide release with the growth of the microbes. Yeah? So those are indirect methods. Yeah? Okay, so that is the methods that you can use for measuring the growth of the microbes in SSF. You can see it's a bit tricky actually, even though this one is, you can say that it's not that really accurate, but that's the nearest or the closest way that we can do in order to monitor the growth of the microbes in SSF. Yeah. And apart from the oxygen consumption or the carbon dioxide production, we can also measure, we can also use other indicators like the production of certain specific metabolites or components. Yeah, for example, certain protein or nucleic acids or glucosamide. Yeah, and ergosterol. Okay, maybe, and also perhaps the products of your interest. Let's say your fermentation is producing cellulase. You take the samples and then you analyze for cellulase. Is there any cellulase produced? So that could be also used as an indicator for the microbial growth. So if there is a cellulase produced and it is increasing, let's say from day 2, day 4, day 6 until maybe day 20, okay? If it is increasing, the cellulase, it means that the microbes are growing well. Yeah? So that is another indirect method for measuring the growth or monitoring the growth of the microbes during the SSF process. So we move to the last factor, environmental factors. So what kind of factors that influence the SSF process? So it's not just SSF. Submerged fermentation pun ada environmental factors yang which it depends on. So in submerged state culture, environmental control is relatively simple because of the homogeneity of the suspension of microbial cells. So it's way easier to control, for example, the temperature of the culture because it is in liquid. So you insert the prop inside it and you can always monitor the growth, the temperature. But for the solid state, it's a bit tricky because it is in solid form. So if you put the things like the prop in at certain point of the substrate, it might not give you actually the full condition, the whole temperature of the whole things, right? So that is the tricky part. So, and because of the homogeneity, it's not actually in suspension, unlike liquid. So that's the tricky part. But still, temperature is one of the important environmental factors that needs to be considered in SSF. Okay. So the critical problems in SSF are related to mixing, heat exchange, oxygen transfer, moisture control, differences in pH, nutrient and products, since the medium is solid, grown with different types of microbes. It's not only the temperature, but also the oxygen transfer, the moisture control, the pH distribution. Okay, you might not know whether the pH at this point is it, I mean, if let's say the optimal pH is 3, so it's very difficult for you to make sure that the whole substrates, the whole solids are in pH 3, compared to if you have a liquid of cultures, you can always stir it, right? So That's the problems in SSF. Yeah? But still, it needs to be monitored. So the environmental factors for SSF include the moisture and water activity. This is one of the important one. Temperature and heat transfer, pH and contamination, and oxygen uptake. Okay, moisture and water activity. So even though it's in solid state, you can see that generally it is dry, but still the cultures need some water in the form of moisture. And moisture is calculated in the form of percentage. Okay, so like you can see here, 30% volume per week. If let's say you have 100 gram of powderized substrate, so 30% is based on that. 30% of, you calculate 30% out of 100, how much? 30 kan? 30. So it means that 30 ml of water to be added into that sort of 100 gram of substrate. Okay, so that's what it means by the percentage. So those who are doing FYP on solid state fermentation, I think there are some of you yang buat solid state fermentation, right? So you have to make sure that you know how to calculate the moisture content, how to calculate the percentage. If it's given like 30% volume per weight, volume per weight tu mesti ada at the end of the percentage. So what does it mean? You have to know that, how to calculate it. Otherwise, your experiment will not be in a correct form. Okay. And what is actually the optimal the moisture level? It depends on the microbes. And certain microbes need 40%, certain microbes need 60%, 70%. So those are the things that are to be figured out for every microbe and for every process, because different microorganisms may have different moisture level, optimal moisture level. Okay. For example, if you see here, for Aspergillus niger, even though it is Aspergillus niger, but if it is grown on different substrates, it will need different moisture level. For example, if it is grown on rice, it needs 40%. If it is grown on coffee pulp, it needs 80%. So even considering the same type of microbe, it has different level of moisture level. Okay. The water requirement of microbes is defined in terms of water activity, AW, representing the availability of water for use and reaction in the solid substrate. So every microorganism, it has its own AW value, okay? And generally for fungi, the AW is lower than the bacteria. So what does it mean? It means that the fungi do not require as much water as bacteria. The water requirement is low. So it means that the fungi can grow on drier substrate, like in SSF. So that's why fungi is very easy to be grown in SSF mode compared to bacteria, apart from the filamentous structure. So the water requirement is low for fungi. Bacteria is high, means that bacteria needs more water in order for it to survive. Okay. Reduction of what AW affects microbial growth, like extension of the lag phase, decreases the specific growth rate, results in low production of biomass. Okay, so I think this is associated with the growth of the fungi. It has longer incubation period. Yeah, the lag phase is longer than the bacteria. The specific growth rate is longer because of the longer growth incubation. Yeah, and it results in low production of biomass. Okay. Temperature and heat transfer. So these are among environmental factors that is also important in SSF. Okay, we know that it's difficult compared to control compared to the submerged fermentation. So how it is to be controlled, how the temperature and heat transfer within the culture is to be controlled. First is by water evaporation, meaning to say that this is example of water being connected. This is the cultures. So when the water is evaporated, the water is provided, and when it is evaporated, it can help to reduce the temperature of the culture. This is normally happen in the industrial place or the pilot scale. We don't have it in lab scale, this kind of practice. Another way how to reduce the temperature and the heat transfer is by aeration. This is for aerobic SSF. So aeration is provision of air. You provide the air, the process is called aeration. Okay, so aeration can also increase the rate of heat and transfer, meaning to say that it can help to distribute the heat, meaning to say that the heat can be reduced. So dia tak panas sangat kalau you provide the air to the cultures. So that's how the temperature and heat transfer can be controlled during the SSF process. pH and contamination. Okay, pH may change in response to metabolic activities. The kinetics of pH depends highly on type of microorganism. Okay, so it's the same thing like the liquid fermentation as well. What is the initial pH that you want to set for the SSF cultures? It depends on the microorganism. Like for the Aspergillus normally is 3. For E. coli is 7, but we don't use E. coli in solid state fermentation. So it has different values every microorganism. And how you can control the pH is a bit tricky compared to liquid fermentation, but it can be controlled by adding certain amount of ammonium salts and urea in the substrate. But I think you have to know first how is the trend of your what is it like the culture, is the pH is going to be reduced or is it going to be increased? So that's the thing that you have to figure out first before you decide to add certain amount of things in order to control the temperature. Okay, so that is pH. So why pH is important? Because it's it is one of the factors that affect the growth of the microbes. So certain microbes cannot grow in acidic conditions, certain microbes cannot grow in basic conditions. So that's why the pH plays an important role in cultivation, including SSF. Kalau liquid fermentation is very easy. Like you insert the pH probe inside the liquid and you can read the pH easily, very easily. And you can even add the acid or base in the liquid form. So for a solid state, it's a bit tricky, but it's still important, the pH. Okay, and then oxygen uptake, oxygen uptake. So oxygen is for those aerobic SSF. Oxygen is one of the important factors, and for the oxygen, normally for the lab scale, for the solid state dalam shake class, it depends on the oxygen transfer throughout the cathode. Itu sajalah. Tapi kalau let's say for the industrial scale, for example the tray or whatsoever the platform, the rotating drum maybe, it is connected to certain it has certain inlets whereby the inlets is connected to the oxygen or the air. So that is how the aeration can happen. Aeration is how provision of air or oxygen by spudging the gas into the cultures. The gas is from oxygen, is either oxygen or either pure oxygen or air. Okay, you provide into the reactor, and then that's how the aeration can happen inside. Okay. So oxygen is one of the important factors for aerobic SSF. For anaerobic, it's not included. Okay, so what are advantages and disadvantages of SSF? SSF leads to higher yields and productivities. So by nature, actually, SSF ni lagi simple compared to the liquid fermentation. Although that you might face certain challenges, for example, in terms of the uniformity, in terms of macam mana nak control the biomass growth semua kan. But the nature of the process is actually simple. You can just ferment the things by having the solid, put the microbes, and it will grow actually. So the nature is simple, and it is useful for certain industries. For example, food lah, tempe, tapai, we can't produce using liquid fermentation. So that's why SSF is needed. So SSF dan liquid fermentation ni dia dalam industri they are needed in various applications, and dia how to say, it's not to replace submerged, and submerged is not to replace solid state, tapi dia diguna depends on the suitability, depending on what kind of application or what kind of products that you want to produce. Okay. So dia ada each of the process has its own contribution and significance. Okay, and for the SSF, because of the nature state of the solid state, okay, it can employ low cost agricultural and agro-industrial residues. So you just chop the substrates and you can use it directly for the fermentation. So that's one of the advantages. And because you are fermenting fungi, right? So perhaps the pre-treatment might not be as difficult as if you want to use the substrates for liquid fermentation. For liquid fermentation, if let's say you want to use sago hampas, you have to do some sort of pre-treatment whereby you have to break first the cellulose, lignin, I think lignin, hemicellulose, and cellulose in order to get the glucose form. Otherwise, you cannot just dissolve the sago hampas in the liquid because it doesn't dissolve. Okay, so those kind of pre-treatment is a bit tedious for submerged fermentation, whereby if let's say you just use solid state, you can just like ferment sago hampas tu dengan perhaps maybe yeast ke atas tu, and then the fermentation can happen easily. Okay, so that is the difference between submerged and liquid, submerged and solid state fermentation. So on that point, actually submerged lagi mahal dia punya process, because you have to break the substrates, you have to use enzymes, or you have to use chemicals, and it's a bit more complex and more expensive. Okay, so that in that point, okay, that's why here SSF, the advantage is it has low capital and operating costs compared to submerged fermentation. Okay, low water volume, which reduces downstream processing steps. Okay, so compared to the submerged fermentation ni bila dia, once the fermentation is completed, you have to remove the water. Actually, you have to do like lots of few things in order to get the final products. So removal of water, you might not see it at lab scale, but at the industrial or at pilot scale, the removal of the water, it is costly actually. So it is a huge step in the downstream processing. Okay, so although that you benefit all the advantages of the liquid fermentation tadi kan, like easy to control the temperature, easy to mix the cultures, but there is a cost at the end of the fermentation whereby it is included in the downstream processing. Downstream processing ni adalah suatu bidang after the fermentation. Fermentation is upstream. So downstream is where you purify the products. You separate the products from all the contaminants. So if let's say the process is requires low water volume like SSF, so it will actually reduces the steps during the downstream processing. So that's one of the advantages of SSF compared to liquid fermentation. Okay, it's a simple process with less effluent release. So I think I've mentioned about it. Okay, simple process, it's about one of the things is because of it doesn't require lots of water, and then the downstream is much simpler than. So it doesn't release effluent is in the liquid form, and so in the submerged fermentation, there are actually lots of liquid waste generated at the end. Even the separation of the products. Okay? So that's advantages of SSF. The disadvantages of SSF, of course, this one lies within the process. The process is solid state is quite hard actually, quite difficult. Even though it's possible, it's still possible, but it's not as easy as liquid fermentation when it comes to scaling up. Scaling up it means that you want to increase the scale of the production. So once, let's say, you have studied in the lab scale, you have identified all the good parameters, the best conditions, and you want to upscale the production. You want to increase, let's say, from shake flask to tray, or maybe the drum, the rotating drum, for you to carry out the solid state fermentation. So there are the whole process, actually, the translation of the scales is not as easy as liquid fermentation. Okay, so that's one of the challenges of SSF, which is in scaling up. Yeah, because when you increase the scale, it means that you have to consider lots of things. For example, the heat transfer, the moisture content even, and the homogeneity. It will not be as easy as you carry out in the shake flask. Perhaps you can just like maybe shake once in 2 or 3 days, but how are you going to apply the things? The same thing is, let's say you apply it on the tray. You have to think like different way of making sure that the cultures are still homogeneous, right? Uniform. So there are those challenges. Okay. The substrate require pre-treatment. Okay, of course, this one actually sama je dengan submerged fermentation. Pre-treatment is you have to reduce the size and everything. Okay, this one is quite subjective. It depends on whether your microbes tu boleh tetap. Kalau pre-treatment yang guna hydrolysis tu is actually kalau submerged pun susah juga. So this one is but pre-treatment is in general is a challenge actually for both. It's not just for SSF. Mixture of bacteria and fungi with different water requirements, as bacteria require high moisture, but fungi needs are lower. That's overall expected. Okay, this one is applicable for a process that use a consortium of bacteria and fungi. Okay, some of the fermentation processes, they don't use only one type of microbe. So they use what we call consortium. Consortium ni adalah suatu gabungan. Gabungan is combination of many types of fungi. Contohnya ragi tadi. Ragi is like a consortium of microbes. It has lots of many types of microbes. So if let's say you want to carry out fermentation in the lab using a mixture of bacteria and fungi, so that's the trickiest part lah. So setiap tu dia ada requirements sendiri. So that's the, and SSF is very synonymous with using consortium of microbes. So that is one of the disadvantages or challenges facing the SSF using the consortium of microbes. And yeah, difficult to determine the biomass concentration. Difficult to know. How are you going to know how we want to quantify? Tak dapat quantify. You just have to observe under the microscope the morphology throughout the incubation period. Whether the filamentous grow longer and longer or not, you have to determine by yourself. And otherwise, you can measure indirectly, like the oxygen uptake. But oxygen uptake pun, that one is only for large scale reactors. For shake flask, you cannot measure the oxygen uptake and the carbon dioxide release. So that's the trickiest part. And perhaps you have to check other metabolites, the products released. So meaning to say that the biomass determination for SSF is not straightforward. It needs indirect methods. Unlike submerged fermentation. Submerged fermentation, you can measure the biomass or the cell concentration directly based on the absorbance. The absorbance measurement is the simplest or the most common method to measure the growth. If the absorbance is increasing throughout the time, it means that, and you can see that from the physical feature of the culture, the keruh, the culture. So it shows that the growth there is a growth. But it's not only the physical feature, that is the one additional thing that you can also see apart from the absorbance measurement. Okay. Don't cite me for that, okay? The most important or accurate way of measuring the biomass or cell concentration is by absorbance measurement. Okay, so that's advantages and disadvantages. Applications of SSF, you can see in the food industry is quite a lot of products produced from solid state fermentation. Okay. For industrial fermentation, there are enzymes produced by SSF. For example, cellulases, amylases, glucoamylases, yeah? Bioactive products like hormones, alkaloids, organic products, organic acid production like citric acid, acetic acid, lactic acid, fumaric acid, biofuel like ethanol. Okay, but these products are possible to be produced by solid state, but it doesn't mean that they are only produced by solid state fermentation. These products are also possible to be produced by submerged fermentation. Okay, so macam amylases, there are amylases produced by liquid fermentation. So you have to understand, this one are some examples produced by solid state fermentation. So in some works, or if let's say you come across articles pun, you akan nampak juga amylases, contohnya cellulase produced using submerged fermentation. Okay. For agro-food industry, traditional food such as Is Ragi, plantain, kimchi, what else? Bread, yeah? Even apa ya? All this cencaluk semua tu is actually a form of fermentation. Cencaluk, tempoyak, right? Because you actually incubate it, you ferment it, then the microbes actually comes from macam kimchi is actually comes from the cabbage. For the durian tu adalah dia punya microbes dalam tu. I do not know what kind of microbes are there, but it's actually a type of fermented food as well. Okay. Other products are like pesticides, polychlorinated biphenyls. So this is in the application of bioremediation, biodegradation of hazardous compounds. Right, biological detoxification of agro-industrial waste like coffee pulp, cassava peels, canola meal. Yeah? These are examples of processes and products produced from solid state fermentation. Okay. So any questions so far? Is it understandable? Guys? No question? Look at that question. Okay, so here are some examples of research articles on solid state fermentation. Okay, how many of you are doing solid state fermentation for your FYP? Is there any? Me? Oh, okay, Debbie. Okay, oh, Lim. Yeah, okay. Quite a lot of you. So you should understand this topic very well, okay? Especially on just now pre-treatment of the substrates. I think that's very, very crucial. And then the moisture content, how to calculate all those things. You have to revise, or you have to learn those things, right? Okay, so this one is the article about vegetable waste, fruit and vegetable waste as a substrate to produce succinic acid using on-site enzyme mixture. Okay, this one I think is using consortium of microbes. So this one is production of lipase from Aspergillus niger using palm kernel cake, soybean meal, fire pith as a substrate. Okay, this one is industrial waste, agricultural waste, sorry. And this one is production of lacase using sago hampas, using solid state fermentation. So that's the end of today's lecture. So basically what we covered today, what is solid state fermentation, how it differs from submerged fermentation or liquid state fermentation, what are the major factors influencing SSF, microorganisms, substrate characteristics, pre-treatment of the substrate, the feedstock, and then biomass measurement, how we're going to measure the biomass, the cell growth, biomass and or the cell growth during the SSF. What are the environmental factors that influence the solid state fermentation? And then what are the advantages of SSF, disadvantages of SSF, and what are the applications of SSF in various industries, the products of Ssf and so on. Okay, any question, guys?